dementia researcher with a blog and a rating.
In a new analysis, my colleagues and I at UCL showed that common genetic variation affecting Apolipoprotein E is responsible for about 3/4 of late-onset Alzheimer's disease. Moreover, we think that 3/4 is an underestimate. The findings imply that if we could eliminate the detrimental effects of apoE in the population, we would prevent most AD from occurring. So what led us to this eye-popping proportion? How do we interpret it? And given apoE's long association with AD, why isn't this molecule's primacy in the etiology of the disease widely recognised yet? First, let's start with a brief primer on apoE. This protein has a very prominent role in cholesterol transport in the periphery, but it is also highly expressed and has numerous roles in the central nervous system. In humans, 3 common forms of the protein exist as a result of genetic variation at 2 sites in the apoE gene. These isoforms are known as E2, E3 and E4. E3 is the most common isoform. About 95% of the UK population have inherited at least 1 copy of the gene that makes it, and about 60% have inherited 2 copies and therefore make E3 proteins exclusively. E4 is the next most common isoform, with about 29% of the UK population carrying at least 1 copy of the gene that makes it, whilst about 2.5% carries 2 copies. The least common isoform is E2, with about 15% of the UK having at least 1 copy of its gene, whilst 0.6% of the population makes E2 exclusively. The E4 isoform is well known to dementia researchers. The genetic variant responsible for it is typically referred to as the strongest genetic risk factor for late-onset AD. Among individuals of European ancestry, individuals who make a mix of E3 and E4 proteins have approximately 3 times greater odds of developing AD relative to people who make only E3. Individuals making exclusively E4 have something in the region of a whopping 12 times greater odds of developing AD. In terms of absolute risk, that is, how likely a person is to develop AD in his or her lifetime, individuals making E3 exclusively have about a 10% chance of developing the disease by age 85. And those making only E4 have in the region of a 60% chance. Conversely, relative to individuals who make only E3, people who make 1 or 2 copies of the E2 protein have much lower risk of AD. This has led to E2 being labelled as protective with respect to AD risk. Individuals making E2 in 1 or 2 doses have less than a 6% lifetime chance of developing the disease. But here we arrive at what must be one of the biggest oversights in dementia research. Since individuals making only E3 proteins are positioned in the middle of a risk scale for AD, between individuals who are making E2 proteins at lower risk and others who are making the E4 protein at a higher risk, E3 is very often regarded as neutral in relation to AD. This view is incorrect. It is akin to saying that moderate smoking in the population contributes nothing to the burden of lung cancer, since non-smoking and heavy smoking influences risk of lung cancer in either direction. This misperception has probably been compounded by the view that if E3 is so common, it cannot be harmful. Regarding E3 as neutral between the opposing effects of E2 and E4 is only a matter of perspective. If we shift our view to that of individuals who make only E2 proteins, anyone else exposed to E3 appear to have precarious positions in terms of AD risk. In a case control study where AD cases had neuropathological confirmation upon autopsy, relative to our lucky few individuals making only E2 proteins, those making E3 exclusively had 7.7 times greater odds of AD. This heightened risk goes to E3 being more detrimental for AD pathogenesis, or because E3 lacks some protective property of E2, is not an important distinction here. Either way, E3 is increasing risk relative to an alternative which is better, at least in terms of AD risk. And not only does E3 confer considerable risk of AD, it is also doing so very pervasively in the population, because it is so common. So what if we were to look at the burden of AD in the population attributable not only to E4, but also to E3? What proportion of the disease occurs in the UK because of these two differences in a single protein? This is where our analysis comes in. We conducted a cohort study in an extremely large population-based resource called UK Biobank, which has genetic information on 500,000 individuals from England, Scotland and Wales. These participants were recruited between 2006 and 2010, then aged 39 to 73 years old. In our research, we used data from a subset of the cohort who were aged 60 or older at recruitment, about 171,000 participants, in order to focus only on people who had appreciable risk of AD throughout the subsequent years since joining the study. We ascertained whether people developed AD over the years since recruitment by confidentially looking at whether AD was coded in their healthcare records or in death registration data until late 2023. This gave us 12 to 17 years of follow-up. We looked at excess risk of AD among anyone producing E3 or E4 proteins, which was 99.4% of the sample. Our reference group consisted of about 1,000 individuals who make E2 exclusively, 0.6% of the sample. On average, individuals making any combination of E3 or E4 were almost 4 times more likely to have been diagnosed with AD in the follow-up period. From the prevalence of E3 and E4 and the excess risk among individuals exposed to these two isoforms, we can calculate the proportion of AD attributable to them, which yielded 74%. To reiterate, this implies that if we had the means to eliminate the excess risk conferred by E3 and E4 to people, with therapeutics for example, we would expect to prevent about 3/4 of AD from arising in the population. Or put differently, if everyone in the UK only made E2 instead of E3 or E4 isoforms, 3/4 of AD would not occur. By extending our calculations, we are also able to partition this proportion roughly into contributions from E3 and E4 separately. We estimated that E4 is responsible for about 45% of AD, with the remaining 30% of disease burden attributable to E3 specifically. So almost 1/3 or more of AD is arising due to the existence of E3, not exactly innocuous. There are various limitations to our study, but the most prominent of these all relate to our incomplete ascertainment of AD diagnoses. Looking at clinical codes among health records misses many cases, and UK Biobank participants haven't lived their whole lives yet. Unfortunately, many more among our sample will develop AD in their lifetimes. Since we know that apoE isoforms affect AD risk, our missing ascertainment will be disproportionately worse among individuals making E3 and E4, and less so among people making E2 exclusively, since proportionally much fewer of them get AD. Since we do not have neuropathological confirmation of AD for cases, we also suspect some cases in our sample may have had other forms of dementia and been misclassified, which would weaken the link between apoE and AD that we observe. So among the reasons why we think 74% is probably an underestimate. To add more context, in some review articles from 2004, an astute clinician called John Wesson Ashford at Stanford University highlighted the circumstances I describe here, and he and his colleagues estimated that up to 95% of all AD may be attributable to E3 and E4. So to summarise, our findings show that if we had the means to target apoE directly, or by modulating factors on the pathway from the protein to AD pathogenesis, we could prevent most AD from occurring. Though our modelling results and conclusion here are simple, interpretation is not always straightforward. For instance, if E3 and E4 are causing most AD, why don't all individuals with these isoforms develop the disease within their lifetimes? Recall the lifetime risk estimates that I provided earlier. Only 10% or so of people who make E3 exclusively develop AD, and even among individuals exclusively making E4, a large minority, in the region of 40%, may not develop AD in their lifetimes. Thankfully, apparent paradoxes like this can be resolved by understanding the nature of complex diseases with multiple causes. In epidemiology, we think about this concept in terms of a spectrum of disease risk, which we refer to as liability. Somewhere on this spectrum is a threshold that, if crossed, an individual will develop the disease in question. An individual occupies a position on this scale as a combination of many factors, genetic and environmental, that affect risk throughout his or her life. If all combined risk leaves someone below the threshold, the disease will not occur for them, whereas if their total risk positions them above the threshold, bad news will be forthcoming. In the case of apoE isoforms and AD, an average individual producing E3 or E4 is much higher up the risk spectrum and hence much closer to the threshold for disease than an average individual making only E2. So it takes fewer other contributing factors to go against them before they cross the threshold. However, despite the relative disadvantage that individuals with E3 or E4 experience with their positions on the spectrum, there is a large range of positions that individuals can occupy on the risk scale due to differences in the multitude of other contributing causes. Many individuals making E3 and E4 won't reach a threshold despite having heightened risk, and they remain unscathed. There is a figure on the blog to illustrate these points. In this sense, it is less accurate to call variation in apoE the cause of most AD, and more precise to call it a component or contributing cause. There may be other ways by which we could reduce AD incidence, aside from interventions targeting apoE structure or function. For example, perhaps by improving education or cardiovascular health in the population. If these are also truly component causes of AD as well, intervening on them would shift individuals down the risk spectrum, and fewer would fall above the threshold for AD. Nonetheless, the fact that there are other factors involved in AD risk does not undermine apoE's position as the predominant cause of the disease. When a disease is multifactorial, not all its causes are of equal strength or prevalence, and as we have seen, these variants in apoE are both a very strong and a very common cause of AD. It is important to realise that without a background of risk from E3 or E4 isoforms, at-risk individuals will be placed so much further down the risk spectrum that all other contributing causes would be much less relevant. Another figure on the blog depicts this concept. This scenario is analogous to the role of smoking in lung cancer causation. Lung cancer has many causes beyond tobacco exposure, which include genetic liability, for example, innate variation in DNA repair efficiency, and probably other environmental exposures like air pollution. Nevertheless, smoking is responsible for the vast majority of lung cancer burden in populations where smoking occurs. If individuals don't smoke, lung cancer is rare. Most non-smokers comfortably tolerate all risks from other sources throughout their lives without meeting the threshold for lung cancer susceptibility. So if we persuade people not to smoke, we prevent most lung cancer. The same will be true of AD with respect to risk from apoE. though the challenge of identifying the necessary interventions here look more taxing than the public health measures towards smoking deterrence and cessation. To conclude, AD researchers are in a privileged position where one protein is key to a large fraction of all disease, and identifying how to obviate the ill effects of apoE is a single research avenue that holds vast scope for enabling AD prevention, and possibly improving treatment of cases too. This potential advantage is not observed for many common complex diseases such as coronary heart disease, where its various component causes are individually much more modest than apoE clearly is for AD. Unfortunately, in the preceding 3 decades, nowhere near proportionate attention or funding has been given to apoE, since the field has focused on other priorities. As a result, among therapeutics currently being trialled in humans for AD, only one directly targets apoE, which is less than 1% of the late-stage drug development pipeline. However, any major progress towards AD prevention may well elude us unless apoE becomes the focal point for our efforts, so it's time to revise our priorities. For most AD, it is inaccurate to say that its causes remain unknown. The most important cause by far is clear, and we need to understand how to modify it. I hope you found this blog post of interest. To read more about this research, please see a lengthy FAQs document that I've posted on my GitHub page, or contact me with queries by email at dylan.williams@ucl.ac.uk.
Thank you for listening. Join the Dementia Research bloggers and share your own views.